All right, please use this to check yourself for quarter 2. Again, check yourself. Please don't just copy. You need to know this for your EOC. So DNA fingerprinting, a thing to remember is this is the gel. When you are looking at what pieces are big and small, remember larger fragments are heavier, so they don't move as far. So this is your heavy fragment or longest one. Whereas down here, you've got your shortest fragment because it moved further. So the shortest piece of DNA, suspect 1, because their fragment moved much lower than all of the other suspects. How many fragments are shown for suspect 4? Here's suspect 4. They have 1, 2, 3 bands. That means 3 fragments or pieces. So who committed the crime? So you have your crime scene DNA bands here. Notice suspect 2 matches 1, 2, 3, all 4 bands. So that means suspect 2 must have done this crime. Here we have an infant, and you're trying to figure out who the dad is. So remember, with paternity, the baby has to match mom or dad for each band. So first it's asking how many DNA bands could have been inherited from the female. So this one, here's 1, 2, 3, 4 bands were inherited from mom. So that means the remaining ones, bands 3 and 5, it's asking could that have been from the female. Here's band 3, mom doesn't have it. Here's band 5, mom doesn't have it. So that means the dad has to have both 3 and 5. So using that, if I look at the remaining males, band 3, male 1 does not have it. He can't be the father. So we eliminate him immediately. Now we look at male 2. Male 2 has band 3. Then we look at band 5. This guy also has it. So that means male 2 would be the father. Now we also use this to catalog endangered species. So this is asking how many bands do species A and X share. So here's A, here's X, here's 1 band, 2 bands. That does not match, that does not, that does not. So that's why it's 2. Which species is most similar to species Y? Species Y is right here. So what you want to do is you want to look at each species, count how many bands they have in common. Species X has 1, 2, 3 bands in common. That's more than any other species. So species X is the most related. So remember, when we're doing DNA fingerprinting, you need to cut the DNA into fragments. That's what we use restriction enzymes for, to cut the DNA. And if we look at this, we have 1, 2, 3 pieces of DNA produced. And the machine that sorts these is the gel electrophoresis machine. There's a diagram of that over here. And remember, then you compare the banding pattern. With genetic engineering, we learned 3 technologies. We learned DNA fingerprinting. That's where we cut the DNA into pieces and sorted by size. Here's a picture of a DNA fingerprint. We learned about the Human Genome Project, where we mapped the genes. Remember, this is really important in gene therapy, where you can find a faulty gene and replace it. And we did genetic engineering, where you add, delete, or change genes, typically from one species to another. So bacteria are typically involved in this for their plasmids. This diagram represents DNA being taken out by a scientist. With genetic engineering, we should know the order. So you cut out a human gene with the restriction enzymes. You put that into a plasmid. Remember, a plasmid is the circular DNA of a bacteria. That bacteria takes in the recombinant plasmid. It's called recombinant because you've combined the human DNA with the bacterial DNA. And now the bacterium is a genetically modified organism, and it makes insulin, which can be used to treat things like diabetes. So here, you're using this diagram for the next page. So let's take a look at this diagram. We have a skin cell, a muscle cell, and a stem cell. Each one has gene A, gene B, gene C, gene D, gene E. However, these red boxes represent genes that are turned on or activated. A skin cell has gene A, B, and D turned on. A muscle cell has genes B and E turned on, whereas a stem cell doesn't have any genes turned on. So keeping that in mind, it's asking if the genes for each type of cell are the same or different. They're the same. Remember, they all have A, B, C, D, E. The red box is the genes being turned on. Do they all have the same genes turned on? No, because remember, this only had A, B, D. This had B and E. This had nothing turned on. So even though they have the same DNA, why are they different cells? So skin cells and muscle cells are different cells because they have different genes turned on. We call having that special job differentiation, becoming the different cell types. The stem cells are different because they don't have any genes turned on. That means they're undifferentiated. So remember, stem cells can become any type of cell. We use that for Alzheimer's and things like that. This one was probably tricky. So here we have the nucleus. You have a functional gene with gene therapy. So you take a functional gene, you find a target cell. That's what it's called. It's a target because that's where it's going. You put it in the target cell, which is typically going to be a virus of some sort. And then that cell will make the functional protein, and you can use that to treat disorders. With inheritance, we learned about Mendelian dominance. That's when the dominant gene determines the phenotype, meaning as long as you have one capital letter, you have the trait. With codominance, you have two dominant genes equally. So you have a roan cow, which is both brown and white. You use two different letters for that. You've got Dalmatians that are black and white. Incomplete dominance is when you have the blend. So red and white make pink. With multiple alleles, there's more than two alleles for a trait. For blood, example is blood type, where we have A, B, and O. And polygenic is when many genes control a single trait, and there's a range of phenotypes. So your height, skin color, eye color, those are all polygenic traits controlled by more than one gene. So this right here is a phenotype because it's physical, it's eye color. Now this is asking, would this organism have a dominant or recessive trait? They have a capital B and a lowercase b. If this is Mendelian, then they would have the dominant trait because they have at least one capital letter. Here are the answers to the Punnett squares. So I included my work. These are ones you're going to want to use paper for. So tall is dominant to short. We have a heterozygote, big T, little t, crossed with short, little t, little t. Half would be tall because half of them have at least one big T. Woman who's heterozygous for blood type O, meaning AO, marries a heterozygous B, which is BO. There's a 25% chance of having A blood, which is right here. 25% chance of O blood, right here. And 25% chance of AB blood, which is right here. We have a pink with a red. What are the chances of being pink? So this is one where you use the primes, or if you are more comfortable using different letters, you can. So to be pink, you can be RW, red and white makes pink, or RR prime, whichever formatting you prefer. To be red, you're just RR. When you do your Punnett square, it's asking about pink. Pink is the heterozygote. There's 1, 2 out of 4, so 50% chance. And this is incomplete because it's a blend. Cystic fibrosis is recessive. A woman who is pure normal, which is 2 capital N's, marries a man who has cystic fibrosis, so that's lowercase n, lowercase n. So in this case, every one of their kids would be a heterozygote, so there's no chance of having cystic fibrosis because the only way to have it is 2 lowercase letters in your genotype. A roan cow, a roan is spotted, is mixed with a white cow. So the roan is the BW, white cow is WW. If you're white, you're WW, so there's 2 out of 4, 50% chance, and this is codominance because you see both colors at once. In humans, hemophilia is sex-linked, normal male, so X capital HY, remember the Y shows that that's a male. There is a hemophiliac female, so she's X little h, X little h. We fill in the Punnett squares. The question is if they have a boy. So I'm looking at only the boys here. There's 2 boys. Each of them would have hemophilia. So there's a 100% chance if you're a boy, you're going to have hemophilia. Huntington's disease is a dominant trait. So a normal man, so if you're a normal man, that means you're little h, marries a woman who is homozygous for Huntington's. That means she has 2 capital H's. What are the chances of them having a kid who has Huntington's? So all of their kids are heterozygotes. Because this is a dominant disease, they have at least 1 capital H, meaning every kid here would have Huntington's. Sickle cell, remember, is codominant, so you use 2 different letters. A man who has sickle cell, SS, marries a sickle cell carrier. A carrier is a heterozygote, so that would be normal, so NS. When we do that Punnett square, it's saying what's the chances of having sickle cell anemia? 2 of the boxes are SS, so 2 out of 4 is 50%. Remember when you're looking at pedigrees, circles are females, boxes are males. Filled in means you have it. This is a dominant pedigree, so that means anyone who has it has at least 1 capital letter in their genotype. Females, there's 5, 1, 2, 3, 4, 5. The male in 1, 2, this male is NN because he doesn't have it. This one, it's asking if it's recessive or just sex-linked. So here I see 1, 2, 3, 4, 5 males have it, whereas only 1, 2, 3 females have it. So because more males have it, that shows me it's likely sex-linked. Carriers can only be females who don't have the disorder, so 4 and 6 are likely carriers. 1 is not a carrier because she has the disease. All right, so remember with the karyotype, to figure out the sex, you look back down here at the sex chromosomes. This person has an X and a Y, so that makes them male. If they had 2 of these X chromosomes, then it would be a female. What chromosomal disorder? We have Down syndrome because 21, you have 3 chromosomes instead of 2. Remember, this is a karyotype. We only use it to detect chromosomal disorders, so an extra or a missing chromosome. So no, you can't detect a gene disorder from that image. Nature versus nurture. So this shows that a lot of traits are genetic because if you're an identical twin, you have a higher correlation, meaning you are more likely to have the same trait than if you are a fraternal twin, which are the same as a normal brother and sister. So diet and exercise is a mix of nature and nurture. You can have good genes, but if you just eat like junk all the time, then your stuff would definitely be very different. The genetic effect of UV radiation. So if you're in too much sun, that could lead to mutations of your DNA. That can lead to cancer. Evidence for evolution. This is when we started going crazy quick. You have the fossil records. So the remains of organisms can be used to determine the age of an organism through radioactive and relative dating. But relative dating just is the order of age. So like this is the youngest, that's the oldest. Radioactive gives you the actual age. Biochemical similarities. The more DNA or amino acid you have in common with the species, the more related you are. So for example, in this chart, a human has lis, glu, his, iso. We're most related to a chimp because they also have the same sequence. Homologous structures are similar structures that may have been inherited from a common ancestor. So this is stuff like a human hand, a cat foot, a whale wing, a whale flipper, and a bat wing. They all have a similar structure, but you use those things for different purposes. And then vestigial structures are things that we lost through evolution, but remain because of a common ancestor. So your tailbone, your appendix, they all used to have some sort of use in an ancestor, but no longer. Evidence for evolution and origin of life. So at first there was no oxygen and no photosynthesis. So the first organisms were anaerobic. They didn't require oxygen. They were heterotrophic. They ate other stuff, and they were prokaryotes. They did not have a nucleus. Then sun, water, and carbon dioxide became available. Photosynthesis started happening, so we ended up with photosynthetic prokaryotic cells that they could make their own food. Remember, photosynthesis makes oxygen, so that allowed aerobic, things that needed oxygen, heterotrophs, to evolve. And finally, the mitochondria and chloroplast formed a symbiotic relationship. We call that endosymbiont theory, and that led to eukaryotic cells. Natural selection and speciation. So you've got this giraffe example here. These are the 4 steps that are involved. So overproduction, in general, there's more giraffes born than can survive. There's already variations. Some giraffes are naturally born with a longer neck than other giraffes, and that leads to selection. The long-necked giraffes have an advantage, that's an adaptation, because they can get food better, they can reach it. The short ones can't reach it, they give up, they die. And if you have a long neck, you're more likely to survive and reproduce. You want a mate who will give you food. If they can't provide anything for you, you're not going to mate with them. And that's why, with the adaptation, more giraffes will have long necks than short necks, because the long-necked giraffes are the ones finding someone to mate with them, and the short-necked ones are not. So the development of a new species is called speciation. This blue line represents a newly formed river that separated the 2 populations, so we call that geographic isolation. And what might have caused them to change is their environments probably selected for different traits. So maybe there was a lot more green plants in this environment, so these guys camouflaged, where maybe there were more trees with bark here, so these guys camouflaged. Now they're 2 different species because if they come back together, they're not going to mate with each other. This we did as a class, but how our systems have changed over time. We went from 2 systems to 6 as far as kingdoms. The evidence we use morphology, which remember is physical traits, biochemical analysis, DNA and amino acids. We use embryology. Remember, everything looks very similar at the beginning. And phylogeny, those cladograms. Levels of classification from most to least. So domain, kingdom, phylum, class, order, family, genus, species. Remember, the way to remember that is Dear King Philip came over for good soup. And every scientific name of an organism is a Latin name made of the genus and species. Humans are Homo sapiens, so our genus is Homo, species sapien. We did this as a class, so I'm not going to talk about it. We did this as a class, so I am not going to talk about it. So here we have the carbon cycle from ecology. What processes remove carbon from the air? That would be photosynthesis. So if you look here, this is carbon dioxide in the air. This is the only thing that's taking it out of the air. Everything else puts it back in. So respiration from plants and animals, decomposition, fossil fuels, all of that puts it back in. Returning carbon to the soil. So into the soil is decomposition, fossil fuels, and fossils. What form of carbon does respiration release? So if we look here, it's carbon dioxide, CO2. And what impact do humans have? We increase the amount of carbon in the air, so we burn these fossil fuels, we have respiration, all of our activities lead to more carbon in the air. Remember, that's bad because that contributes to global warming. This we did together in class, so I'm not going to go through it. All right, this graph is predator-prey. The predators, basically, when you have too many predators, your prey goes down, but when your prey goes down too much, then your predators start creeping up and so on. And when you look at these, your prey is always going to have higher numbers than your predator. Competitors are biotic, they're living factors that limit the size of populations. These are different relationships. Some of this we didn't talk about, but you could have used common sense for this. So parasitism, you have a host, the host is harmed, so a tapeworm steals nutrients from the intestine of a cow in which it lives. Mutualism is when everyone's happy, they both benefit. So these fungi provide water and nutrients for trees, and trees provide carbs for the fungi to eat. An example of communication is ants use pheromones to create trails from their nests to food that other ants follow. An example of being territorial is fighting fish like betas will use gill flaring to scare away competitors. So here we have carrying capacity is on the left, so population goes up and then it fluctuates around carrying capacity. Remember, carrying capacity is the maximum amount of organisms an ecosystem can hold. There's only so much space for everyone and resources. Limiting factors are things that limit the population. So it's asking what might be a limiting factor for trees. So the amount of sunlight would be, they need the amount of space, carbon dioxide, because plants need that, soil, that sort of stuff. Current human population is to the right. Even with COVID, our population keeps going up. And how might disease affect the growth curve? So it could cause it to level off. Currently, that's not happening, but too much disease could cause it to level off and reach carrying capacity. These things are stuff we didn't mention, but I was hoping you were familiar enough with these from the past. So imprinting is when a baby duck follows their mother and copies everything she does. So ducks imprint on the first thing they see. Ataxis or tropism is something like a plant growing towards the sun. Migration, butterflies travel long distances at certain times of the year. Geese migrate. Classical conditioning is Pavlov trained dogs to salivate when they heard a bell because he would bring food with the bell. And then when they heard a bell, they assumed food was coming and they'd start drooling. Habituation, think a habit is something that forms over time. So after repeated exposure with no harm, gorillas will allow humans to approach. Human impact. So acid rain is caused by nitrous oxide and sulfur oxide from factories. Water pollution is runoff from land. Climate change is excess greenhouse gases like carbon dioxide. Biodiversity loss is from deforestation, cutting down trees, and urbanization, so building stuff. So burning fossil fuels is associated with climate change and too many greenhouse gases. Stability is impacted by deforestation and urbanization. All of this decreases ecosystem stability, and that's biodiversity loss. In the lakes and streams of the mountains of North Carolina, lose species because of water pollution and runoff from land from nearby cities. Human impact. So if our population keeps increasing, there will be a rapid depletion of natural resources, so we'll run out of resources. The transgenic organism to clean up oil spills is bacteria, so we can genetically engineer bacteria for oil spills. And if you have a non-native species, an invasive species, like kudzu, which is prevalent here in North Carolina, then that native species has to compete for resources with a non-native species and the ecosystem becomes unbalanced. Things like kudzu take over entire ecosystems. Then this is talking about biomagnification. So biomagnification is when concentration of chemicals increases as you go up the food chain. So you notice as you go up, each one gets more of the chemical, whereas bioaccumulation is just within one organism. So bioaccumulation is the pesticide within a fly itself. And then this is really our earth science. So if the bluefish gill suddenly began to die and the water had a lot of nitrogen and phosphorus cause toxic algae blooms, and that's due to something called eutrophication. And what that does is that disrupts the nitrogen and phosphorus cycles. And the cause of this is runoff from fertilizer because fertilizer has a lot of nitrogen and phosphorus. All right, that is it. Good luck on your EOC. And again, if you need help with anything, please let me know.